Can I confirm that you see my screen?
We do see your screen. Yes. Thank you.
So I thought since the last two talks were both on inflammation, that we might start there while it's all fresh in everyone's mind. And my first question is pretty old at this point, but I want to ask it again because I think the field is changing rapidly. Do you think of ME/CFS as an inflammatory disease? And I said during the science days that I was concerned that inflammatory was not precise enough. And what I've seen from the talks is that precision is being added to our study of inflammation. And so, one particular point that I think it was either um, Vanessa or um, Michelle who made is that chronic inflammation is different from acute inflammation. And I would like to ask the question of those who are interested in this particular topic. In what ways are the two different? Are there different cell types? Are there different molecules that are involved in chronic versus acute inflammation?
I can maybe start and then I'll probably throw it over to Bob or Vanessa. But from my perspective, I think there are similarities and differences. There are very similar cells that can be involved, but like Vanessa and I were both talking about, there are some key cell types that are the first to the scene, and they are really involved in these acute situations of infection, inflammation, and can rapidly come and go and change their phenotypes to respond to things as quickly as possible. Now, the markers on these cells, I think, can change over time. And it really depends on the context, where you are in the disease, uh, what's happening, what kind of disease, and what other kinds of insults are there. So I do think that, yes, your innate immune system responds first, and you have key cell types there, and then your adaptive will come in later. But there's still your innate immune cells involved in chronic inflammation as well. They just may have different markers and different functions at that point. But I'll hand over to Vanessa and Bob and others to comment.
I mean, Michelle said it pretty well explained. Yeah. So I think the cell players are the same, right? They just have been in a toxic environment or are kind of confused as to what are the next steps. So then they start behaving improperly. Yeah. So, and that can cause secretion of biochemicals that are different that were not supposed to be there, were not intended to be there at that point, but now they are being produced. So
Well, Navia, what's your thought on this chronic versus um, yeah, acute inflammation?
Yeah, but I'd say, I mean, classically, medicine has defined acute inflammation according to, four characteristics: this rubor, calor, dolor, and functio laesa. That basically, redness, heat, swelling, and loss of function are the classic signs of acute inflammation. In order for tissues to have that and be able, neutrophils. I mean, one of the we're just really fascinated by how far the mitochondria go in establishing the functional capacity of all cells. Like sometimes, in order to make a cell the first responder to a new site of injury, you actually have to have fewer mitochondria so that there's greater capacity for glycolytic support for chemotaxis. And for the ability to rapidly develop an oxidative burst. And you can imagine, just in the same way that red cells don't have mitochondria because we'd be bad to have an oxygen carrying cell with an organelle in it that was consuming oxygen. You know, you don't want a first responder cell to have a lot of mitochondria in it that would, you know, diminish its capacity to respond, you know, briskly to microbial pathogens. But then the real thing is turning that off. Okay, so and that's where we keep getting back to these cycles of metabolic, you know, change that have to occur in order to, you know, resolve an inflammation. And that's where we're really stuck. I mean, ultimately, that's where people are stuck. It's not an acute inflammation. They're stuck in the resolution of the full process. And there are a lot of, you know, receptor changes that occur, and even sensitization of cells to, you know, specific signals that change from the acute to the chronic state. And we're learning more and more about that. But I think these models and the PET tracers are really going to help guide us toward understanding the rainbow of the inflammation. I mean, you know, so just in the sense they're up quarks, down quarks, strange quarks, you know, there are, you know, many different flavors of inflammation, and it does us a disservice to keep, you know, we're forced to use the same words because we don't have, you know, well-defined new words to talk about the gradients from, you know, from a classical acute to different forms of chronic inflammation. But we'll get there. You know, it just requires, you know, good science and persistence and new tools.
Got it. Braden, have you thoughts on acute versus chronic inflammation?
I completely agree with everything that has been said, but would also just sort of bring back attention to the idea of priming as well. There may be cells floating around that are not actively involved in inflammation, but that are ready to begin or participate in inflammation irregularly with abnormal signals, kind of turning on and off as part of a chronic illness as well. So that's something to be considering and looking at.
Thanks. Okay, let's move on to this other question. How are inflammation and mitochondria linked? It's clearly TSPO is part of the answer to that, but I'm not sure how the signal is transmitted from other cells to the mitochondria of a cell that's responding. And so, is this purinergic signaling, Bob?
So cholesterol ends up being a rheostat for establishing the sensitivity of cells and mitochondria to environmental stresses. So the more cholesterol should not be in mitochondrial membranes. Okay, it's just in a healthy cell it doesn't happen. When, you know, however, a cell that has been, you know, involved in chronic stress responses for, you know, some time, more cholesterol is deposited in these mitochondria associated membranes, the MAMs, you know, between the mitochondria and the ER, and that kind of creates this, you know, metabolic synapse, and that's what it is, you know, that allows for calcium to rapidly, you know, be transmitted from ER stores into mitochondria that then, you know, you alter the function. So we think of, you know, TSPO as dialing up, you know, the sensitivity of mitochondria to, you know, inflammatory signals, and ultimately it's all about lipid membrane remodeling that is instantiated by, you know, by TSPO, and there's some other things too, but that's as good as another one. Another one that I'm kind of interested in, I'd just throw it out now, is you know, what happens when more and more cholesterol comes in, it ends up crowding out other kinds of lipids in the cell membrane. And so, there can be a loss of, ceramide can be ceramides are play a role interacting with cholesterol to establish these membrane lipid rafts, and it'll be important to sort the role of sphingomyelin, ceramides, and cholesterol, and how those three interact.
Yeah, I was just going to ask about rafts. So my impression is that the proteins that accumulate in lipid rafts care about the composition of those rafts. And so you actually have proteins that are going to move in and move out depending on how much cholesterol you have, for example.
Yeah, well, so a lot of receptors act as dimers, and so when a monomer is present and not near another monomer, that let's say it could be IL-6, so the receptor for IL-6 could in a monomer is not really that makes that cell not very responsive to IL-6, but when the monomers are brought together in a lipid island, this microdomain of a membrane lipid raft, then that receptor becomes able to bind and to signal, so it effectively increases the sensitivity of the cell to cytokine signaling by having an increase in membrane lipid raft microdomains that will patch and cap actually with cell activation.
Thank you. Okay, so Michelle, how does TSPO respond here? Is TSPO only a mitochondrial membrane protein?
That's a great question. And I mean, Rob has already sort of painted the picture of what we think that TSPO is really doing at that level. And so you asked the question here, what is the sort of origin story of TSPO? So maybe I'll give a little bit of information there because I think it is interesting. I hinted at it in my talk, but it was discovered in the kidney. And it was binding diazepam, and so people thought it must be like the central benzodiazepine receptor. And so that's why it was called the PBR. And it was known as that for a long time until a bunch of pharmacologists came along and started really probing it with different pharmacological tools and doing all sorts of genetic modulatory tests. And basically what we found was it is not related to the CBR at all. It's its own distinct entity with its own functional roles. And one of those roles, like Bob has been hinting at, is to do with cholesterol. It is translocating cholesterol from the outer to the inner mitochondrial membrane, and it's playing a part of a sort of a protein complex that helps assist with that process, which is one of the rate-limiting steps of steroidogenesis. So folks have been trying to understand its role there. It doesn't seem to be essential for that, but it is definitely needed. It's not the only player, but it certainly has a big role in this modulation and translocation of cholesterol. So I think that may indeed it sort of help emphasize what Bob was saying with its ability to dial up and down the sensitivity of the mitochondria to external stresses. So it's like this little sensor. Yeah, very, very interesting. Is it only found there? Predominantly. I mean, I think that there will be some instances where you'll find it elsewhere, maybe on the cell membrane, but it is predominantly on the mitochondrial membrane. So I don't know if that helps a little bit.
It does, that helps a lot. I
Yeah.
want to know how the tracer gets there.
That's another great question. And I am a chemist at the core. I love cooking and making things that are helpful, both in the kitchen, but also in the chemistry lab. And one of my biggest passions is trying to get something across the blood-brain barrier and across cell membranes, across all these barriers, so that we can bind to and understand and have an effect on different markers of interest, whether they're PET biomarkers or pharmacological. So how do we get these tracers to that place? Careful chemical design and a lot of headaches. But we do have to very carefully design these molecules such that they have the right physicochemical properties that allow them to cross multiple membranes. And that means they have to have the right lipophilicity, so they can't love lipids too much. They need to love them just enough to get across, but not to get stuck. So we need to tweak these little pharmacological and physicochemical properties, sort of tuning the different properties of the molecule such that they can get through all these barriers, but still bind very specifically to that target. So I showed one of the chemical structures, and I won't bore people with all the chemistry, but suffice it to say, there is a design there. There's a careful design, and it is a lot of fun, but it's a big challenge to try to design something to get to where you want it to go while maintaining the specificity for your target of interest.
Yes, it's clearly a really important and difficult problem. Is the part of the tracer that binds to the target usually modeled on an agonist for that target, or something that is already known to bind?
This is also an important comment that you're raising. So typically what we want is an antagonist or something that doesn't have an effect at all. Actually, that's the most perfect situation. And just for everyone online, and hopefully there's a lot of people still here, a PET tracer is called a tracer for a reason. It's given at trace amounts, very, very small amounts. They need to be below where we would get a pharmacological effect. So imagine taking an aspirin tablet, lick it, throw it away. That's like the dose you're getting for a PET tracer. So you're not going to have any pharmacological effects by licking an aspirin tablet, I don't think. Maybe a tiny bit on your tongue. But that's my analogy to say that it is the tiniest amount that we give. We're not occupying all the receptors. We're looking at just a very small subset, actually, a representative amount, because we want to be innocent bystanders. We want to be quiet observers of what's going on with the biology that you want to study. So we want to be quietly looking at and not impacting or affecting that receptor or process.
Exactly. Classic principle of tracer kinetics.
That's it. You know it.
I grew up in tracer kinetics.
I know that. Yes, Rob.
Okay. So let's see. Last question on this list for inflammation has to do with neutrophils. And honestly, the kind of inflammation that Bob Navio described earlier is the kind that I used to teach in cardiovascular physiology in medical school. And we didn't talk at all about neutrophils. We talked about the fluid movements, edema, the changes in permeability, all those things that make the classic symptoms of inflammation. So, Vanessa, are neutrophils always involved in inflammation?
So they can also spontaneously migrate. And they've shown this in cancer as well, and in vitro, where they don't necessarily need to have a chemotaxis, so like an inflammation agent that's calling them to the tissue. Yeah, so they're able to migrate into tissues without a particular, chemokine calling.
That's interesting.
And then there's also reverse migration, which is another one that's still being studied. So they have the ability to
By
move.
that you mean that the neutrophil can come back into the blood?
Yeah, like they'll be in an inflammation site and then they'll crawl out back to the bloodstream. So they were involved at some point in inflammation, but now they're getting out of it.
Right.
Yeah.
Okay. Thank you. All right. I suggest, I want to make a quick announcement that although I was really keen on asking questions of the Decode ME crew, Joshua Dibble was called away from his desk and is unable to participate in our discussion this afternoon. I'm planning on sending all the questions that I carefully built for him to his email address, and I will hopefully be able to get them out to all of you, 393 people, by some mechanism that I can't predict at the moment. Okay, so let's move on to the next discussion. This is on theories and drugs. It's been said a couple of times at this meeting. I know Bob Navio said it. I might have said it because I strongly agree that a good way to test theories of ME/CFS is by using each theory to pick an FDA-approved drug and get a clinical trial going. And so the question that I'm asking here is, for your theory or your favorite theory, what drug would you choose? And so I'm going to ask Bob Navio first because I'm pretty sure I know the answer.
Yeah, well, the only one that I have available at the moment, just because this is an area that's new to the world's pharmacopoeia. I mean, no pharmaceutical chemist has really had the incentive to make, drugs that inhibit ATP signaling. So we have just one that was created by accident 100 years ago, Suramin, that, has been used to treat African sleeping sickness for 100 years, but in the late 80s was found to actually possess non-selective purinergic antagonist, properties. So it would inhibit a variety, half the purinergic receptors, half of the 19 purinergic receptors. So that's the one that we would put to test first. But we, if that study is successful, then we would hope it would really incentivize pharmaceutical chemists to start creating a shelf full of more selective agents that are available by mouth.
Do these drugs that are under development or would be under development serve as antagonists for those receptors? Is that the goal? Inhibitors?
Yeah, yes. So there are, the adenosine receptors in general, well, there are 4 different kinds of adenosine receptors. In general, they tend to be the coda that, so if ATP signaling is the chemoattractant for neutrophils and it starts off the inflammatory process, once inflammation becomes resolved, that ATP is metabolized to adenosine, and then adenosine binds to inhibitory receptors that actually stop, say, okay, resolve. And so there's work, in the purinergic, pharmacologic development world to develop antagonists, of the P2Y and P2X receptors, but agonists, of the adenosine receptors.
Cool. Okay. I wanted to make the point that the zebrafish model and the mouse model that Dr. Oliveira and Dr. Peterson talked about are not yet models of the disease. They are models of hypothesis, in particular the attack-and-age shunt hypothesis. And I wanted to ask the question of the group, can useful questions be answered by testing animal models with candidate drugs, even though those models aren't yet genuine models of a disease, because we don't know that the attack-and-age shunt is operational in ME/CFS. In other words, might Suramin even help a zebrafish that is sick because it just has human ACOD1 expression?
I'd say yes. Yes, well, I would say, there's a chance but that's a deeper discussion because one of the interesting things about inflammation is that in addition to, a direct effect on, let's say, ACOD1, it's also inhibiting this iron sulfur cluster dependent protein like aconitase. So when aconitase is inhibited, it also increases the substrate for the the decarboxylase that produces itaconate. So in other words, if you prevent aconitase from being converted to isocitrate because iron sulfur proteins are oxidized, okay, then there's more connotate around for ACOD1 to work on and to make, it's anyway, the whole cycle of the TCA or Krebs cycle, and knowing what not only the anaplerotic inputs are, but the cataplerotic out out points, and of course ACOD1 is a cataplerotic enzyme that siphons carbons out of the Krebs cycle. But anyway, that sometimes a knockout won't reproduce the full aspect of
it's entirely possible. And sometimes I have to admit, I think I'm studying a subset of the cell danger response and the healing cycle. I often feel that way. So how about other
It's a
people?
good way to feel. It's a good feeling.
You think so? Okay, I'm not surprised. I wondered if we could build an animal model that's more like the whole cell danger response by implanting an osmotic mini pump that just establishes a steady state of extracellular ATP.
Yes, I think that is, a really interesting idea that could prove the whole hypersensitization, concept, because you could regulate the amount of ATP that's being produced, and you'd have basically regimes, specific doses that the mice would receive, and there'd have to be some kind of, behavioral challenge, some kind of challenge, to see how because it's really what keeps on coming up in my mind is that in the same way that TSPO is a rheostat for the inflammatory sensitivity of a cell, I think that purinergic receptor expression and then downstream signaling cascades can become sensitized so that the animals can have essentially a crash in response to a relatively minor let's say physical stress, or we'd have to work out what the paradigm for the stress model would be, but that's a very interesting concept. Suramin is kind of a strange drug because it has a very long half-life. So if you do one injection, one IP injection in a mouse, it's a 7-day half-life. In humans, it's a 2-week half-life, and then if you give it for a few times in a row, it'll go up to a 2-month half-life. Okay, so it's a little bit different, but this whole ATP sensitization story is one that's very interesting. You could get there in the 90s. There's a literature from the oncology world, both from the Netherlands and then from UCLA, that actually did continuous IV infusions of ATP to determine what the maximum tolerated dose was and what kind of side effects there were and that kind of thing. And so that could be used to potentially guide this mouse mini pump, experiments that would be interesting.
I'd like to have those references if you can send them to me.
Sure, sure, but what I can see immediately with the mouse TP model is, you know, give them to Michelle James, okay?
Exactly.
And look at
That
what the
was my next
TSPO, the signal, that PET
We're
tracer
gonna
looks
send
like.
mice to Michelle.
Yes,
Yeah.
we've talked about that. I'm happy to receive any mice and quickly test them. And actually, I wanted to ask you all what you think about some of these long COVID mouse models, and could they be any help as well? Or
I don't know about them. How are they built?
well, they're inducing a COVID infection and
Oh, so it's
waiting.
directly.
Yes, and waiting an amount of time and looking at fatigue. And I don't know enough about them, but I just started to look into it. I know they're mouse models are fraught with a lot of limitations. And that's why I do love with PET, we can go directly into people quite rapidly. But when it comes to testing therapies, it may be helpful to have a couple of models up our sleeve to narrow things down, right?
Is Toto Oliveira on the call still? No.
No.
Okay, so I'm going to ask Randy to try this. Um, what do you think about this notion of testing hypotheses? Obviously, you've already invested in it, so you must at least think it is worth trying. But does it have a precedent? Are there other drugs where the development has been based on a mouse model or a zebrafish model of how you think the disease works?
That's a great question, Rob. I'm sure the answer is yes. Whether I can come up with a great example off the top of my head is another question.
If you can tell me anytime in the next month, I'd really like to know.
Okay.
So in general, though, Randy, the the mechanisms that you've used in zebrafish models to develop drugs are situations where the target has already been determined, right?
Well, either the target or the cause. I think there are in fact very powerful tools for finding new mechanisms that reverse the disease symptoms when you have an assurance that you have the right cause, that the construct validity is there. And I think the challenge that we face in this particular case is, as you very well articulated, Rob, we have a model of ACOD1 expression. We don't know if it's actually a model of ME. And so even compounds, for example, the ACOD1 inhibitors that seem to be offering improvement to these animals, it could simply be because they're inhibiting the cause of the symptoms, not because they would also be useful in a patient with ME. So in some respects, compounds that work through a different mechanism, not by inhibiting ACOD1, might be even more interesting because they might more generally be able to address these symptoms without directly targeting ACOD1.
So for example, there's a molecule called P110 that was described by Daria the other day, I think on day 2 of the science meetings. And honestly, I'd never considered it until now, the possibility of testing it in your zebrafish model of ACOD1, because it could be the downstream of the attack-and shunts is a mitochondrial fragmentation phenotype.
Mm-hmm.
And it could be that that's actually causal. And if the drug can revert mitochondrial fragmentation, then maybe it would help. And maybe the fish phenotypes would improve.
Yeah, I think that's a very interesting idea, certainly testable. And I think we could both look in the model of ACOD1 expression and see if there are mitochondrial changes. And then if there are, we could absolutely test P110.
Yeah, just to point out the balance aspect of this is that a lot of the DRP1 antagonists that the things that inhibit mitochondrial fragmentation and then kind of naturally push things more toward keeping mitochondria fused have been beneficial in short-term models, but lead to what's called stress-induced hyperfusion. When carried too long. Okay. And stress-induced hyperfusion results in actually a paradoxical, increase in the cell sensitivity to apoptotic death with a stimulus. We see it in Parkinson patients. And actually in the brains that we've looked for, this in adults with autism, it's been a similar issue, the stress-induced hyperfusion, or SIH. Yet drugs like P110 typically do show benefits early on.
Thank you. So I'm going to push this question even further to David Systrom. You've been using some drugs that were helpful in ME/CFS, at least conceivably. And should we be testing those in models that whose mechanism may not be the mechanism of ME/CFS?
Well, I think a great question, Rob. I'm not a mouse person. I'm aware of the differences between human and mice. However, I know there are some really fascinating adoptive transfer models in mice where plasma or even immunoglobulin from affected patients with ME or long COVID are given to mice, and there's a phenotype that looks kind of like ME and fibromyalgia for that matter. So I think translational research, just generally speaking, of course, is the holy grail, going back and forth between models and the human. I think the other approach is the one that Wenzong, I think, put out there, and that is we've got tons of FDA-approved drugs out there. We know a lot more about potentially viable biologic pathways to these diseases, whether it's upstream or downstream autoimmunity, inflammation, neurotoxicity, mitochondrial dysfunction. And I think the alternative approach is to go to a place like the Broad, which has a vast library of all the drugs ever created in the world with AI infrastructure, and in that setting, kind of do what Wen has done to a degree with his large survey, and that's to determine if there's anything out there that suggests drug X may work or not. But I do think I would echo everything previously said about the utility of using repurposed drugs, because as everybody in this big room knows, to get a drug from the start to the end is millions and millions of dollars, and how many years. So our patients are crying out for answers. I think both approaches are great: translational medicine models into the human, using repurposed drugs, and surveys such as what Wen has put out.
Absolutely. Okay, I'm going to move us to oxygen, your favorite topic.
That's my topic, Rob. Just
I
making
believe that's your favorite topic.
making certain you're looking at me. So,
Yeah.
well, I think some broad statements, perhaps. We are very intrigued by the notion of exercise intolerance and related symptoms in both ME and long COVID being related to either oxygen delivery problems or final oxygen uptake and utilization problems, or both, and the relationship between the two. So on the delivery side, we have, and I mean the royal we here, we have evidence of dysautonomia and what I think is left to right shunting. We have data out of the Boost lab in Amsterdam using muscle biopsies that get capillaries of basement membrane thickening, a barrier to oxygen offloading. We have evidence of red blood cell abnormalities and their interaction with the muscle capillary bed and inappropriate vasodilation and vasoconstriction as a result of abnormal red cells, too big, too stiff, and their metabolism may be off. So we have all these things that could lead to deficient O2 delivery, even at a microscopic level in the muscle and perhaps elsewhere. This isn't to, for a moment to forget the CNS and other places, and even the peripheral nerves. On the other hand, we have what I have learned from Dr. Navio, and that is, and from the Voos lab and our own work, that we have a potential problem with a mitochondrion. There's an intrinsic problem with a mitochondrion, and that's either number or function or both. So that's an area likely deserving of different types of treatment. You folks were just talking about those things. So O2 delivery, O2 uptake, and utilization, the problems are not mutually exclusive. They may both exist. They may be related to each other. So I think we need to continue the work in this area.
You have a screen in front of you that has my questions on it?
I do.
I
So
wanted to know if VO2 decreases with ME/CFS severity. Do you know that?
I would emphasize my wheelhouse is VO2 max. That's maximum oxygen uptake at peak exercise. And I do not want to comment on resting O2 uptake. And I'm told all the time by patients, wait a minute, doc, my problems aren't exclusively with exercise. I feel lousy at rest as well. So what's going on there?
Right.
But I will defer to others about metabolic studies done at rest. At peak exercise, yes, generally speaking, the VO2 peak declines as a result of, or associated with severity. And then you hit on I promise I didn't read bullet number 2 in advance. So resting VO2, right. Don't know. Don't know. I'm going to plead agnostic on that one. And then O2 consumption and ROS formation. Yeah, there is a delicate balance. Dr. Navio could speak in a more educated way to this than I, but both hypoxic conditions and hyperoxic conditions can lead to a condition where there's increased ROS formation and then therefore are mitochondrial poisons. So there's a balance there, and it may not be the same in everyone. I'm aware of pending ongoing studies of hyperbaric oxygen, of supplemental oxygen, and then there's another thought that even brief bouts of hypoxia might be helpful in some patients. Wide open field, Rum. Great question, so.
Wenzong, I had trouble arranging questions for you because I didn't know the content of your talk. And I wonder if you have some thoughts about all the drugs and the supplements that were tried by patients in your survey that we should be thinking about in the context of our experimental work.
Excellent question. Sorry, I'm at the airport. I hope the sound works.
You're doing great.
Okay, great. So a couple of things that I think I'd like to mention. One is, it seems like different treatments that patients self-reported so far, as I mentioned earlier, affect individual symptoms. So then the question is, is different patients with different symptom profile going to be really helped by any given or a combination of treatments? So that's one question that I asked myself, and I don't know the answer. That's what we're trying to, to analyze further computationally. But you obviously can have two answers. One would be it doesn't. In that case, that would suggest that we're fixing the symptoms, individual symptoms, but not really addressing the underlying problem. I think that's one explanation that could be the situation. the other one would be yes, right? So then you would get, let's say, a subset of patients are going to be significantly helped by a specific treatment or treatment combo. You know, in that setting, then it gets to a question that you asked earlier, you know, is it, you know, do we actually have a universal mechanism for this disease, or different subset of patients have different, you know, underlying mechanism?
Right. You know, I've always thought that the cells that are affected determine this distribution of symptoms. And cells affected, in my view, are the ones that were infected by the triggering virus and their neighbors, because those are the ones that will be exposed to the consequences of the attack on H shunt. Bob Navio's theory is much more general and is made chronic by a high level or a sustained level of extracellular ATP.
Signaling. So that's the key, is they become more sensitive to even, you know, low levels of ATP. So that's a critical distinction that we've measured in autism, you know, that I think also applies to ME/CFS and relates to this, you know, sensitivity to relatively minor stresses.
Is this a ATP concentration that's in the micromolar range? Extracellular?
It's still in, yeah, so it's still in the micromolar range, but what happens is that the receptor expression is changed, and then the amplification of the signal post receptor ligand binding is also amplified. So it's, and when we specifically look at you know, ATP itself, it's acutely increased, like you know, after a stress in autism, we can measure it to be elevated in xanthine and hypoxanthine and inosine, and you know, so in some of those different purines we can measure 2 days after a stress, but within 3 to 5 days it's back to baseline, yet the response of the cells lasts for a month after the stimulus.
So that's why the increased sensitivity has to be a feature of the disease.
Yes, I think so. And it's not necessarily the basal concentration. Well, I'll just say it's not the basal concentration of ATP.
I understand. Yes. The receptor is acting in the absence of what we would normally think of as a effective stimulant.
You're right. It's amplifying the cellular response to that signal. It's, you know, this happens in Dower worms as well. Nematodes, you know, they upregulate a lot of chemosensory neurons in their cuticles. Okay, you know, yep. and part of that makes a lot of adaptive sense because they have to be responsive to changes in the environment in order to survive.
Perfect.